This is going to take us through a brief review of Unit 2.3 Eukaryotes. So eukaryotes are about 10 to 100 micrometers in size, much larger than prokaryotes. They are more complex, they evolved later, they contain a true nucleus as well as other organelles, which are membrane-bound internal cell structures. They divide by mitosis, as can be seen in this little GIF right here, which involves splitting the nucleus and the cell. They can be unicellular or multicellular organisms. This includes animals, fungus, and plant cells, each of which have sort of unique features. Here you can see a fully labeled eukaryotic cell with all of the cell parts that you should be familiar with. We'll practice drawing one of these in a moment. But notice that they each have individual functions which become important. The nucleus contains the genetic material of densely packed DNA. The smooth endoplasmic reticulum has enzymes for drug detoxification. The flagellum, if present, would move the cell. The rough endoplasmic reticulum contains enzymes like ribosomes, which would synthesize proteins. These ribosomes can also be freely floating in the cell. They are 80S ribosomes compared to the 70S in a prokaryote. They have the cytoskeleton, which is made of different tubes that kind of support and change the shape of the cell. Mitochondria produce ATP in cell respiration. Centrioles will assist in mitosis. The cytoplasm, once again, is a liquid that contains enzymes for metabolic reactions. Lysosomes contain enzymes like lysozymes, which help to recycle old cell parts. The cell membrane controls what enters and exits the cell. The Golgi apparatus takes proteins made by the ribosomes and ships them out of the cell after it has modified and packaged them. These proteins would likely be coming from the rough endoplasmic reticulum. So we're going to practice drawing a eukaryotic cell here quick. I would recommend just start with the cell membrane, a large structure. Go ahead and give it a flagellum. Once inside, we've got a nucleus. You can also provide a nucleolus. Outside of that, give it a little wobbly structure and give it some dots. That would be our rough endoplasmic reticulum. You can do the same over here, but don't give it dots. That would be our smooth endoplasmic reticulum. Then you can go ahead and start labeling structures like the mitochondria, which you'd want to provide the internal structure there too, the double membrane. We also need to have lysosomes. You can give it some centrioles, which look like two cans that have sort of been tipped, one on their side. This can also have the cytoskeleton coming out of it. We need a Golgi apparatus. And this is going to cover most of the structures, except we also need some ribosomes freely floating on the inside here. Let's see if we got everything. Yeah, I think we pretty much covered it. All right, good times. So, obviously those need to be labeled, and you might also have to write what the structures do in there as well. Plants are different because they have a cell wall as well as chloroplasts and a vacuole. They have almost everything else that animal cells had, except they often lack a flagellum, lysosomes, and centrioles. These objects have their own functions, which I won't bother reading here. You should be able to identify structures in an electron micrograph, and the process of elimination here really helps. The largest structure is going to be the nucleus. Any small kind of bendy structure on the outside is likely going to be the endoplasmic reticulum. Besides that, anything that's highlighted you could assume is some sort of small cellular object. So here we've got a lysosome. A mitochondria is going to be pill-shaped. You might be able to see the internal structures there as well. The Golgi apparatus will likely be a little further out than the rough endoplasmic reticulum. We've also got 3 ribosomes shown here, just floating around. The cytoplasm would be a non-labeled portion. The cell membrane is, of course, the outside of the cell. And so, again, by using the process of elimination, you can narrow down most of the objects here. Extracellular components would be components of the cell that are on the outside of the membrane. The cell wall is a really good example of this, as well as other objects that have been discussed before, including cell structural objects like cell membrane proteins, such as glycoproteins shown here. Comparing and contrasting plant and animal cells is a good idea, looking at what organelles they have. In addition, plant cells have these objects called plasmodesmata, and they store starch, whereas animal cells store glycogen. Animal cells have some different objects as well. They have cholesterol in their cell membrane as well. and they are mostly rounded in shape, whereas plants are going to be a little boxier. And to compare prokaryotes and eukaryotes, their size is different, their DNA shape is different. The DNA itself is going to be more naked in a prokaryote, not wrapped around histone proteins. The size of the ribosomes is different, their cell division type is different, and the cell parts can be called organelles, which you can name a couple examples of. Boom.